Hi everyone, I'm Zhao Yi, and I'm a fourth-year plant biology student. The title of my experiment is Identify Homozygous UBR7 Insertion Line in Arabidopsis thaliana. The objective of this research is to identify the tDNA insertion knockout lines in order to characterize them for their response to pathogen infection. In this presentation, I will display my approach and data on screening homozygous UBR7 insertion line in Arabidopsis. UBR7 is a putative E3 ubiquitin ligase that coordinates with proteasome to degrade proteins inside plant cells. In our previous study, we identified that UBR7 also interacts with a nucleotide binding leucine rich repeat NLR protein, NNLR, in Nicotiana benthamiana. NLR class of immune receptors recognize viral suppressors of RNA silencing and are important in plant immunity. Nicotiana benthamiana that express NNLR protein is resistant to lethal tobacco mosaic virus due to hypersensitive response. A hypersensitive response is a defense mechanism that involves rapid cell death that at the infection site as well as the surrounding tissue during a viral infection. Rapid cell death traps the virus inside the infection site and prevents further cell-to-cell viral transmission. In Nicotiana benthamiana, this response is turned on by the N protein. Because the response leads to rapid cell death, the NLR protein is tightly regulated and only becomes active during viral infection. Our previous study showed that UBR7 participate in the regulation of N protein. When there is no TMB infection, UBR7 forms an unstable complex with NLR. During TMB infection, P50 helicase domain protein of TMB associates with UBR7 and release it from NLR complex. More stable NLR activates the hypersensitive response. To investigate whether UBR7 has similar regulatory function in Arabidopsis thaliana, we first identified the tDNA insertion knockout line in order to characterize them for their response to pathogen infection. To determine if a plant contains homozygous UBR7 insertion, two sets of PCRs are conducted for each line to check if the sample has wild-type UBR7 gene in test set 1, and if the sample has UBR7 gene with tDNA insertion in test set 2, using the primer listed on the slide. The predicted genotyping result outcome is displayed in the figure. Wild-type sample should display positive result in test set 1 and negative result in test set 2. Heterozygous individuals should display positive result in both tests. Homozygous individual should display negative result in test set 1 and positive result in test set 2. For line 036382, 7 samples and the control display a positive outcome in test set 1. No sample display a positive result in test set 2. Based on the result, we know the sample number 4, 5, 8, 9, 10, 11, and 12 in this line are wild type for UBR7. No tested individual contains homozygous or heterozygous UBR7 tDNA insertion. For line 205844C, no sample display a positive result in test set 1. 6 samples display a positive result in test set 2. The test result shows sample number 2, 7, 8, 10, 11, and 12 in this line contain homozygous UBR7 tDNA insertion. No tested individual is heterozygous or wild type. Insertion of a large piece of DNA disrupts the transcription and translation of coded protein. In Arabidopsis, heterozygous individuals contain a set of functional UBR7 gene and a set of disrupted UBR7 gene after tDNA insertion treatment. Homozygous individuals contain 2 sets of disrupted UBR7 gene. Consequently, they are not able to produce functional UBR7 protein. The line allows us to study the effect of losing UBR7 with and without pathogen infection. Based on data on screening of homozygous UBR7 insertion line in Arabidopsis, no tested individual is homozygous in line 306386. In line 205844C, 6 tested individuals are homozygous and will be selected for future study in Arabidopsis thaliana immune response to various pathogens. Here's the reference, and thanks for listening to this presentation.